All right, let's jump right in. We're talking about one of the biggest hurdles in treating cancer today, getting a fast, accurate genetic diagnosis. For so many patients, this is step one, and it's a critical one, but it can be painfully slow. We're going to break down some new technology that's poised to change all of that.
Just think about that for a second. Imagine you're waiting for news that could change your entire life. Now, what if you could shrink that wait time from 3 weeks down to a single day? That's not a fantasy. It's the real promise of a breakthrough coming out of new research from the Children's Hospital of Philadelphia.
Okay, so to really get why this new tech is such a big deal, we have to start with the problem itself. And that problem is buried deep in our genetic code. It's a specific kind of error, a faulty blueprint that can literally tell a cell to become cancerous.
And that problem has a name. It's called a gene fusion. Think of your DNA like the world's most complicated instruction manual. A gene fusion is what happens when two totally separate genes break apart and then get stitched back together in the wrong order. This creates a brand new, scrambled instruction, this hybrid gene that basically screams at a cell to grow and divide out of control.
So finding these fusions, it's a huge deal. They're not just the troublemakers that can cause cancer. They're also like bright, flashing signposts, what scientists call biomarkers, that tell doctors exactly what they're up against. And even better, they can be a perfect target for highly specific drugs. We've seen this with the famous Philadelphia chromosome in leukemia, and with drugs like entrectinib that go after tumors with what are called NTRK fusions.
So if these fusions are so important, how have we been looking for them? Well, for years, doctors have relied on a set of tools that are pretty dependable, but they come with some serious limitations.
The gold standard for a while now has been something called short-read sequencing. A perfect example is the CHOP Cancer Fusion Panel. It's a clinically approved test that specifically hunts for fusions in 119 genes known to be involved in cancer. You can think of it as the trusted detective that's been on the case for years.
But here's the catch. There's always a catch, right? This method has some major trade-offs. Sure, it's great at finding the fusions it's designed to look for, but it has big drawbacks. It's slow, mostly because to make it cost-effective, labs have to wait and batch a bunch of patient samples together. And maybe even worse, it has a huge blind spot. Because it's only looking for those 119 known targets, it can completely miss brand new or really complex fusions.
And all of that brings us to the real-world cost of those delays. 14 to 21 days. That's not just a number on a slide. For a patient and their family, waiting up to 3 weeks for a clear diagnosis, for a plan, well, that can feel like an absolute eternity.
But what if there was a better way to read the code? Researchers are now using a totally different kind of technology. Think of it like swapping out an old magnifying glass for a powerful new lens. And here's where you can see the night and day difference. Short-read sequencing is like trying to piece together a story from a book that's been put through a paper shredder. You're working with these tiny 150-letter-long snippets. But long-read sequencing, that's like getting to read whole pages. We're talking over 10,000 letters at a time. You get the full picture, the full context, which makes spotting a weird, scrambled instruction, like a gene fusion, so much easier.
So how do you prove your new tool is actually better than the gold standard? The researchers at CHOP designed a really clever two-part experiment. First, a control test. They used it on samples where they already knew a fusion existed, just to prove it could find them. But then came the really exciting part. They used it on cold cases, samples that the old test had already checked and declared negative.
And this is where the story gets really interesting. The team focused this cold case hunt on gliomas, which are a particularly aggressive and hard-to-diagnose type of brain tumor.
Now get this. They took 24 glioma samples that had already been through the best-in-class short-read panel. The official medical verdict on every single one of them was no fusions found. As far as anyone knew, these were diagnostic dead ends. But then they ran them through the new long-read method. And in those negative samples, they found and confirmed 20 new potential cancer-driving gene fusions. 20! These were critical genetic errors that had been completely invisible to the standard method.
And you don't have to take my word for it. The researchers themselves put it perfectly, saying the new tech uncovers complex rearrangements in samples deemed fusion panel negative. This wasn't just a small step up,
It's like they flipped on a new kind of light and suddenly saw things that were hiding in plain sight the entire time.
So what about that control group? The samples where they already knew what to look for. The results there were also incredibly strong. Out of 24 positive samples they tested, the new long-read method correctly identified the known fusion in 19 of them. That's a fantastic success rate. It proved the new method isn't just good at finding new things, it's also highly reliable and sensitive.
So what's the bottom line here? What does all this breakthrough science actually mean for doctors and, most importantly, for the patients they're trying to help?
Well, just look at this. This timeline says it all. The old way? 2 to 3 weeks. With the new long-read tech, a full analysis can be done in under 2 days. And for a single, urgent case, you can get an answer back in less than 24 hours. This isn't just an improvement. This is a complete revolution for diagnostics.
And this powerful new lens also gave us deeper insights into the biology of these cancers. It found that the high-grade gliomas, the more aggressive tumors, had way more of these gene fusions than the low-grade ones. This gives more weight to the idea that more genetic chaos can help fuel a more aggressive cancer.
So let's bring it all home. This really matters because it means less waiting and less anxiety for patients. It means finding real, actionable drug targets in cases that used to be a complete mystery. It gives doctors a much more complete genetic blueprint of the enemy they're fighting, which opens up incredible doors for personalized medicine. And it can do all this from the original tissue sample.
This is, without a doubt, a huge leap forward. But it also makes you wonder, right? As our tools for reading the fundamental code of life get faster, cheaper, and more powerful, it really begs the question, what other medical mysteries are we on the verge of solving? And just how much is medicine about to change? Thanks for joining us.